Thank you, Rupert, for hosting me again today, and thanks everyone for being here. So I'm Thomas Diefenbach. I'm at the Ragon Institute of MGH, MIT, and Harvard in Boston. And the Institute was formed, just by way of background, the Institute was formed originally with the task of developing either a cure or some kind of therapy for AIDS, for HIV infection. And in the past 2 years, we've also been very active working on COVID-19, of course, and we have also groups that work on tuberculosis. We have a BL3 facility for that, and also some ancillary work that's done on things like Zika virus and so on. So we're quite active in infectious diseases in general. We have a sister institution in South Africa, in Durban. So as a group, we're interested in, of course, in looking at the immune system, looking at immune cells and phenotyping them, and looking at the infectious process in patients infected with AIDS. And the challenge of this is, of course, the number of markers that are often required. For example, in approaches using flow cytometry, which is what immunologists normally use to address these questions, at least for cells in blood, is that they would be using panels of markers anywhere from 15 to 30. We have a BD Symphony that looks at 30 marker panels. So in tissue, if we were to look in tissue, for example, we're very interested in looking at the reservoirs of the virus in tissue or at sites of infection. It's, of course, more challenging to look at that many markers in tissue. And so naturally, the question is, how does one approach immunofluorescence from a multiplexing standpoint? What are the options, and and in this way I'm going to present to you my own journey in this process in terms of the evolution of how I try to add more markers and add that capability to our imaging facility for our users. With that, I'll talk about the technology, at least as far as in the past, there have been approaches, things like multispectral imaging and just the process of multiplexing itself. But those traditionally have been limited in terms of throughput, because they're usually, you take a single field of view at a time, and you might do some multispectral imaging on that and do some unmixing. And that can be very time consuming. And what we require is to be able to see globally the entire tissue and to be able to see where cells are going in, for example, lymph nodes, and profile them in terms of their activity during infection. And so a slide scanning system or format is quite amenable to that, because you can have higher throughput, you can look at many slides at a time, and you can look at many fields of view and stitch them together. As you'll see, I'll show many examples of this. Stitch them together so that you have sort of a Google Earth view of what's going on in the tissue, and especially if you're looking for things like rare events among different cell phenotypes. So with that in mind, many years ago, so we started using the TissueFAXS system about 12 years ago, and then it became important to add more colors. And so at the beginning, we had this simple setup pictured here with two cameras, a bright field and fluorescence, and a motorized. This has a motorized stage, as most slide scanners have. There's an 8 slide insert on the stage, so we could do 8 slides at a time. So at the time, we had the LumenCore Spectra X light engine, which is an LED light engine that offers 6 LEDs. And we had the optional near IR 730 nanometer excitation wavelength. So that afforded us the ability to add one extra color to our palette. So most microscopes are limited in the number of colors you can image. Typically, you'll have a DAPI channel, so the blue, and then you have a green, red, and a far red. So FITSI, rhodamine, and Cy5. So we added that 5th color. And my goal was to add, because the turret on the microscope could take 10 cubes, so I thought, well, okay, we could do 8 to 10 colors. How could I do that? And so I worked with Chroma to try to develop a combination of filter sets. So just using generic filter sets, combining Alexafluor dyes with brilliant violet dyes. And this was basically the system that we had on the scope at the time. We used a quad band filter set for the DAPI, FITZI, rhodamine, and Cy5. And then, as you can see listed here in these different positions, different filter sets for either Alexafluor or brilliant violet dyes. So this gave us the ability to look at between 8 and 10 colors without having to do multispectral imaging, and we would be switching back and forth between the cubes. This actually worked out fairly well, and we still use these cubes in this system for just the basic imaging, the straightforward imaging. This is an example of a scan that we did of tissue, and you can see how it's like a montage of many fields of view that are stitched together. And here's an image showing human colon. So a lot of our tissue samples are biopsy samples from patients with AIDS. So we have here human colon. On the left, just an overall view of the stitched region, and then some close-up images of... So usually we're imaging at 20x, and that is pretty good for resolving even, well, certainly single-cell detail, but even some subcellular detail. So because our camera is 2048 by 2048 pixels, so it's a very good resolution camera. Here's an example of lymphoid tissue, and this is a 5-color immunostain. So the 5th color, because we're using that 740 nanometer diode on the Lumencore Spectra 3, we're imaging Alexafluor 750. So looking closer, the purple color here, so these are all the cells in different colors, color channels. The purple ones are the AF750 cells, and Alexafluor 750 is really nice because it's very bright, good quantum efficiency. There's very little background. So if you have sort of a problem with some background or autofluorescence, well, maybe not autofluorescence, but more background, AF750 is quite a good addition. You can add it as a 5th color without having to make any changes, large changes to your protocols. So that's a nice addition that we had. This is a 7-color immunostain, in this case of lymph node. I'm not sure if it's from mouse or marmoset, but you can see how we're starting to build up the color palette. Here's just a close-up of a region showing on top left the some of the brilliant violet dyes and Alexafluor dyes that we were using. So at least 7 to 8 color staining with this approach was fine, and I mean it takes a bit more time, of course, because we're switching between cubes to do this. Here's an example of a breakdown of when we're going beyond 5 colors, we get into the Alexafluor dyes, and this is an example of a 6-color stain with the black and white raw images on the top. The problem with brilliant violet dyes, of course, they're excited in the blue, like DAPI, and then they emit in different wavelengths depending on their type, is that DAPI itself will tend to show up in all the brilliant violet channels, except maybe brilliant violet 785. Really depends on the sample prep. So instead of DAPI, we're using, as you can see on the top left here, TOPRO 3, which occupies the Alexa 647 channel, and then that helps us to use whatever brilliant violet dyes we like to employ. So that was fine for a while, but then TissueGnostics thankfully came out with a multispectral addition to their microscope, and you can see it as this large orange housed camera on the front top of the microscope, the front port. So we added this system to do some testing and try some imaging. We also combined it with an updated light engine, so the Lumencore Spectra 3, which is an 8 LED light engine, and we also employed a 747 nanometer excitation in the far red in place of, I think, the YFP, the 510, because we didn't really see a need for that often, or at least I didn't see a need that often for a YFP emission range or excitation range fluorophore. So the multispectral approach is different in that we're actually sampling, and on the bottom left here you'll see sort of a schematic, a TissueGnostics schematic of what's called a lambda stack. So we're sampling at 10 nanometer intervals across the emission range of that fluorophore, and in the top right here we're seeing the profiles of the emission, and we're able to profile them, and then we use single stain controls to get those emission profiles, and then in the StrataQuest software we're able to mathematically separate them. So this is nothing new. I mean, people have done this for years and years with confocal systems, like the the multispectral detectors and so on. So this kind of approach with lambda stacks has been used before. But in this case, we're using it in a slide scanning format and a sort of a mild high throughput format to cover very large tissue regions. So the profiling is also useful for if you have just a slide without staining, you can profile the background fluorescence or the autofluorescence if you have older tissue or if you have tissue that's prone to having difficult to work with autofluorescence. You can profile that and then actually subtract it out using the software as well. Here you can see the interface that we have in the TissueFAXS software. Up on top we can see 8 of the slides. Well, 4 slides were loaded in this case out of the 8 positions. And then we have now on most microscopes, if you were to see the control interface, you'd only see 4 filter sets, right? DAPI, FITSI, rhodamine, and Cy5. Of course, this system is rather unique, I think, in the world because we have our old setup and then we have a slightly newer, the multispectral setup. Going left to right, if you'll bear with me, we have the transmitted light path, we have a DAPI filter set in blue, we have a brilliant violet 785, then the Alexafluor 750 in the purple color. I had difficulty choosing which colors to use because at some point you're saying to yourself, well, what should I use? So then we have the brilliant violet 420, brilliant violet 570, brilliant violet 650 going left from right here. Then the Q-Cy5. So that's part of the quad band dichroic filter cube. So there's a Q-Cy5, Q-Texas red. The Alexafluor 488 cube is just left over from the original setup where we might want to check with a single band cube whether our quad band cube has crosstalk between the DAPI and the FITSI channels. Then we have the Q-FITSI. So all these other cubes on the right here, that starting with the letter L, these are part, and TissueGnostics has a different cube setup than this. This was just the original setup we had. It's 2 quad band dichroics that are covering most of the visible bandwidth on the emission side. So basically we would be switching between these 2 cubes and sampling at 10 nanometer increments across the emission range of that particular fluorophore. We don't necessarily have to sample the entire range. We could just sample that part of the spectrum that the fluorophores emission would fall. This is an example here. On the left you see an image, what it would look like if we were imaging in multispectral with 7 or 8 colors, and it does look a bit like a mess, but you can also see on the right here. Now I'm told that the interface for StrataQuest has been updated from this. This was already a few years ago. Like 4 years ago, I think. And so, but in this case, we have separate buttons so that we could access any particular 10 nanometer increment for any of the different channels or fluorophores that we looked at. So we can see visually where the peak might be. Now, of course, this is an unmixed image. So we haven't done any spectral unmixing or separation of the different channels. Here's an image of a lymph node, and I'm going to be drawing your attention to the left ROI or region of interest here, outlined in the rectangle in a later slide. So we're going to be looking at that. This is, again, is still, well, it's mixed, I suppose you could call it. It hasn't yet been unmixed. If we were going to look at a single fluorophore, like Alexafluor 647, on the left here you can see an example of a spectrum. In this case, we sampled across the entire spectrum, which normally you wouldn't do, but I just wanted to check the entire spectrum on this in the initial testing, and you can see one large peak corresponding to the emission of the Alexa 647. We also have an autofluorescent channel here for background. If we're looking at something with 7 colors or 8 colors, this is an example of what you would see in the software. You'd have these different marker spectrums, and then your list of fluorophores on the left here, including the background fluorescence, which you would have from you could either identify it from what you know is background in the tissue, or it would be identified from a slide that just had not been stained with any antibodies, either primary, well, they could be a primary, but no secondary, let's say, for as a control. And then there's also an autofluorescence channel. So in the middle here you see the mixed version, or before unmixing, and on the right there's an unmixed version. Now in this particular version we didn't get all 7 or 8 colors. It was maybe 5 because a couple of the antibodies just didn't work in this preliminary test that we did. So, I was going to say that it's nice to be able to then look at many colors and separate them spectrally, but we wanted more colors. As I mentioned before, we're using panels of antibodies in flow cytometry to look at anywhere between 15 and 30 markers in blood. And so to do this in tissue, we need to employ multiplexing. So we need to employ a method to be able to basically stain the tissue and image it and then wash off or use heat or whatever to remove the antibodies or the fluorophores at least, and then restain and re-image to build up a map. One method would be to use the opal dyes, and the opal dyes from PerkinElmer, they basically are washable. Well, they're not washable. They use heat basically to remove them. So you can go through multiple cycles of staining and imaging and heating to remove them. Now that heating, it's been reported, can cause some damage to certain types of tissues. So I read recently about beta-mercaptoethanol being an alternative approach to washing away the opal dyes. But in any case, this is an example of a 6-color opal stain. And so it would be like opal 520, 540, that sort of thing. And you can see the spectral profiles in the middle. So, okay, so the opal dyes were working. I was curious to test to what extent we could separate them because I had heard from, now for years there's been the nuance camera system, which is basically a multispectral system that works quite well. And I'd heard of a report of a group in Germany that imaged a sample that had Alexafluor 488, GFP, and FITC all in the same sample together. And they were able to mathematically separate or unmix those, even though the overlap in the emission is very similar. So I thought, well, opal 520 and opal 540 are about a similar kind of test. So I wanted to see if I could separate those effectively, because then I could use them as well. So here's an example of tissue that was stained with a marker for B cells and T cells, and these markers would always be exclusive from each other. So what we're seeing is the mixed version. So this is before unmixing, and I'll be drawing your attention to the square here, the ROI here, where I'll show you a before and after. Now this was a very preliminary test. We just did it quickly, and it's a little bit out of focus. Normally it would be sharper, but just to show you on the left here, the mixed version prior to unmixing. Now we have green and red fluorophores, or at least we're capturing black and white images, but of course we're assigning color lookup tables to them, but we have either green or red. Now on the left you can see some yellow, of course, because there's overlap in the mixed or prior to unmixing. On the right you can see that the green and red are faithfully separated. So, and spectrally you can see it here represented on the right. On the top set of curves you can see in the green, the large green curve is the opal 520. The next row down, the next set of curves with the red large curve, that's the opal 540. So these are very close in terms of spectral overlap on the emission side, and they have these secondary curves as well, but we were able to separate them completely. We also had Alexafluor 647 in the sample just as a control to make sure there wasn't anything amiss with the system otherwise. So we had employed this multispectral imaging approach to a study of COVID-19 back in 2020, looking at basically the lymph nodes from postmortem patients, and looking, because it was found that their germinal centers had been disrupted for those that were terminally ill with COVID-19. And so we undertook a study, and this was a Cell paper, that looked at basically the distribution of B and T cells in germinal centers in these COVID-19 patients. And so basically what happened is, with this germinal center disruption, there was a reduction in the number of BCL6 positive B cells as well as T follicular helper cells. And that was accompanied by an increase in TNF-alpha production. And so this is just the model from the paper on the left side, showing on the top that normally the germinal centers form, like a very distinct distribution and organization, whereas in COVID-19 there's a reduction in the BCL6 cell differentiation, increase in TNF-alpha production, and then a disruption of germinal center formation. And so you can see it histologically, even in bright field images. There's like, whereas you look early on in the disease, there's very little disruption or in control patients, whereas in those that had succumbed to COVID-19, there's quite a distinct absence of germinal centers in the lymph nodes. And the lower panels here, just from the paper, showing the reduction in the BCL6 positive cells. On the left, the late process of the disease. On the right, those that were controls. And these are just close-ups. So this is a control specimen, and then in COVID-19, of course, you can see they're either absent or they're just more diffuse or disrupted in their organization. So having utilized some of those techniques, in particular just using a standard filter cube set to increase the number of colors, or using multispectral imaging, the next step, of course, would be to multiplex. That is to do multiple passes of staining and imaging and repeating those passes to build up a map. Here's a schematic from UltiView. They have markers that are, they use barcodes that are washable. And so on the left, you can see the staining step, and then you can introduce your fluorophores, and then you can do some imaging, and then do some washing, and then image again. And then you can iterate this to create basically, you're imaging the same piece of tissue over and over again, but with different panels of antibodies to build a map. And that's represented down here with the codex image. So, so one thing you need is software that can take those images captured at different times with different panels of antibodies and overlay them and then register them, like line them up. Right, so that process is schematically shown here, where the StrataQuest software has an ability to do what's called tissue registration. So you might have two images of the same tissue with different staining panels, and the software can align them. Now, this is a kind of a gross alignment. There's also something within the software called cross-correlation, which will align the tissue at the single cell level, which is what we needed to have that kind of precision. So we did a test recently. Again, this is lymphoid tissue. This is showing the StrataQuest interface. And here we see the entire tissue section that we were interested in. And we imaged this twice. And we did two staining panels on this. So it was basically a DAPI with just 3 fluorophores. We wanted to keep it simple at the beginning and just have a 3-marker panel for each scan. So the ROI that I have here outlined in the rectangle in somewhere near in the middle of the tissue, we'll be focusing on that region and zooming in on that region. And I'm going to show you, so this is actually 2 images here that you're looking at. I'm going to be showing you the before and after of the cross-correlation process. And on this little inset here, we can see the fluorophores with the little eyes. Those eyes just indicate whether the channel's on or off. So we have DAPI, Sci5, Texas Red, FITSI channel, and then we have DAPI P1, Sci5 P1, Texas Red P1, and FITSI P1. So those are just the P1 is the panel 1. So the way this works is that you'll open your first scan, and then you go to add channel. It's called add channels, and then you can just open up the second experiment, the second scan, and then add those channels. Of course, everything has to be the same. You have to have the same exact region, and the slides can actually be taken out, from the stage insert and put back in with quite a bit of precision. The Merzhäuser stage inserts are made so that even when you're manually taking out a slide and putting it back in, it should be within 5 microns of where it was before. And I haven't yet experienced any difficulty with rotational effects. So this process here of alignment works in the XY plane and not so much in like rotational aspects. So this might look a little bit disconcerting. This is that zoomed in ROI or region of interest I was telling you about. And this is before the cross correlation. And so this is just all the channels. I know it looks like one of those old 3D glasses where the glasses are on backwards, but okay, so this is just the overall, all the channels together. Here we're looking just at the DAPI channel. So this is prior to cross correlation, and so you can see, so I've made the two DAPI channels green and red. So all the channels, like DAPI1, DAPI2, FTSE1, FTSE2 will be green and red. So that's this is before alignment, this is after alignment or cross correlation. And here's the FTSE channel before and after. So you can see that alignment was quite faithful. And then we have the Sci5 channel before and after, again before and after. And here's the Texas Red channel. Of course, those weren't the fluorophores. This could have been AF probably 568. Sorry, I don't have the list of antibodies that we use, but I can provide it if someone needs it. This is before and after. So you can see even in the very fine structures, I don't know if these are dendritic cells or what, but even in the very fine structures of the cells, the extensions, you can see that there's very good alignment. So this makes it possible then for us, and we could have added a third pass. We could have done a third panel of markers and then build up this map, and it's actually unlimited. We could have done 50 or 100 markers in this with the same technique. And then once we've done this in the software, we can then apply all the tools that StrataQuest offers that Rupert has probably been talking about as far as the quantification of these cells, either in terms of their distribution in tissue or their association with each other or their marker profiles. So yeah, so having talked about some of the approaches that we've used to look at multiple markers in tissue, I wanted to talk a little bit more about the capability of the slide scanning systems and other applications that we've used and the throughput. On the left here, we have our old Zysilira PS1 confocal. So as most confocals, it usually is quite laborious and time-consuming to image slides and on a confocal microscope, because you're sitting there for hours and you're looking at one slide at a time, one region at a time. TissueGnostics, thankfully, came up with a system that utilizes a slide loader. You can see it on the right here. This large box here is a slide loader that can hold up to 120 slides at a time, and it's combined with the spinning disc confocal scan head. So basically, with this system, we have been able to increase throughput quite a bit and combine it with confocal imaging and combine it with additional colors. So we could have employed this tissue registration approach with the system as well, if we wanted to. Of course, being a spinning disc confocal system, it's very fast. You can do Z-stacks, and I was the first to show that you can use oil immersion on this and you get automated oil immersion, say, of cell cultures, because all of the slide loading process is horizontal. So you don't have, you can pre-add the oil drops to the slides and then it just autofocuses perfectly fine. Here's an example of Marmoset lymph node. This is a confocal image and it's actually a Z-stack that was taken with the system. And I'm not going to show any like Z planes or close-ups of in particular, but just to show the image quality. And this particular image was from Daryl Irvine's lab at MIT, one of our group members. And this is just a close-up showing the lymph node germinal centers, either in medulla on the left or cortex on the right. Just if you were curious to look at the distribution of cells in tissue, the StrataQuest software also lets you look at cell gradients and, in this case, perhaps the polarity of these cell populations in different conditions or different parts of the tissue. This is an example of the workflow that you would see in the TissueFAXS confocal. On the far left here, you can see the map of the slides in the 3 magazines. And so we were doing what's called a job, is just basically a run of many slides. You can assign different camera settings or configurations to different slides. So you could actually run 2 or 3 different experiments from different users in the same session. And usually we let these run overnight or even over the weekend. And because of COVID-19, a lot of the times we had people working from home, so they would just use TeamViewer and could run the system while they're at home or remotely. Then we can see to the right of the slide map, we can see examples of slides, the previews of the slides. So we usually would have 6 to 10 lymph node sections per slide. And then on the right, you can see just one of those images. This would be a confocal image taken of that particular lymph node. When it does the Z-stacks, these Z-stacks aren't usually too big, but you can step through the stacks. And so you could use software to look at them in 3D if you like, but of course we're only looking at cell populations in 2D, so the point is just to be able to get some of the depth as well in confocal. This is an example of a group. My background is actually in neuroscience, so I was hired to basically help this group of immunologists to get into imaging and look at the cells and manipulate the cells. This is an example of a group from MIT that's looking at brain tissue. And it's quite powerful for that too. I mean, look at how many sections you can get on a slide and the kind of throughput they had. So it's quite on the opposite side of the spectrum, as opposed to looking at things globally over very large regions or looking at very large populations of cells and correlating them. You can also look for very rare events. So in this case, this is a group many years ago looking for pancreatic stem cells, so stem cells in pancreas. And the green here that you can see, this is the TissueQuest software, by the way. The green that you can see is a single pancreatic islet. Blue is, of course, DAPI, and the red spots are stem cells. So out of maybe like 200 or 300 islets, you might see one stem cell. And so in this case, we were scanning many, many pancreatic tissue sections trying to find and characterize these very, very rare events. I've had cases where we have different methods to localize cells, of course, like flow cytometry and so on. Sometimes we have T cells. I mean, there was one study where we had T cells where we were looking for a phenotype that was 1 in 10,000 cells. So it was extremely rare. And so we had scanned, and we were trying to use confocal, but it didn't work. And we used the image stream, it didn't work. They were just too rare. So we basically put the cells, all of the cells, like 2 million cells on a cover slip, and then scanned many cover slips, and we did it at 100x oil immersion, and we got about 200 cells from that particular scan. So it was enough for the paper. So even really rare events, you can take advantage of this platform. Now, looking at cell culture in more detail, this is a nice example of a large field of view. This is, of course, a tiled scan. So these are human macrophages that were infected with HIV. The green cells are just normal cells. They haven't been infected, and they're taking up the virus naturally, as they would. That's their job. The red cells are those that are actively infected with HIV. And I'm going to be direct, and this is a 63x oil immersion Z-stack, actually. And this is just a small part of a very, very large scan. But we're going to be zooming in on this group of red cells over here on the right, in the middle. We'll be zooming in on those. So here you can see that group here now in the middle, a little bit closer up. And if we move into this cell pair in the center, the large red cell and the green cell, we see here the cells close up and in the Z, showing the Z-stack. So the task or the goal of this particular study was to look at the fate of the HIV virions or particles in the cells, in these macrophages, in association with particular endosomal compartments like RAB5, EEA1, RAB11. And so we were able to create these composites or projections of the cells from the Z-stacks, and also look for events such as this, where there are cells, there's one large red cell engulfing a smaller cell. Depending on the strain of HIV, you will get these macrophages forming these very large multinucleated monster alien cells. So it's quite unusual. We had to actually be selective in terms of the strain of virus we were using, because sometimes these cells would just be so huge. I mean, they would have like 20 nuclei, and they weren't really the kind of cells we were interested in. But yeah, so the nice thing too is that the StrataQuest software and even TissueQuest can look at quantify the puncta, or the points, these spots within the cell. So you can try to associate localization between HIV particles and the different endosomal markers. Just another close-up of that cell. So I won't spend too much more time, but just to show a little bit of the capacity of StrataQuest and TissueQuest, for that matter, to segment cells within tissue and give us the ability to identify them. This is lymph node. In this case, this was a study looking at PD-1 receptor and PD-L1 and PD-L2. They looked at lymph node and they also looked at tonsil. Here's human tonsil. So we have basically CD8 cytotoxic T cells being segmented here. They're outlined in the red. And then we have the PD-L1 ligand associated with tonsillar crypts. This is back to lymph node. Just another example of the segmentation power. So here we have ICOS, so T cells being segmented in red, GATA3 population, IL-5, so the T helper cells, which aren't as common. And then, so going back to that study, the COVID-19 study, where we were looking at the germinal centers and the BCL6 positive cells, we're also looking at association of B and T cell conjugates. So the additional power of the StrataQuest software is to be able to not just look at the cells, their markers, the intensity of the markers, let's say the area of the cell or the distribution of the cells, but also and you can basically create any kind of application, as it's called, to, in this case, look at cell-cell contacts. And so we conducted this study to look at just how often you saw T and B cell conjugates within germinal centers or outside of germinal centers. And so we even moved further, and this is actually just showing the workflow within StrataQuest. This is and I'll just go through this quickly. This is the original color overlay. And then if we go further, this is just the DAPI channel in black and white. This is the AF750 channel. This was a 5-color stain, so we have AF750 as well. This is the Cy5 channel, Texas Red channel, the FITSI channel. This is the AF750, Texas Red, and FITSI. So we're looking at like combination of markers. Here's the segmented nuclei and CD4. Here is probably easier to see. Now we're looking at just the cell masks of triple positive CD4, IL-4, NCXCR5 cells. Here are the CD19 cells, the masks of those cells. These are just the seeds, what they call seeds of the CD4, IL-4, CXCR5 cells. So this is the overlay, and there are actually, it's maybe hard to see, I'll show it closer, but the cells that are touching, so those two cell types that are in contact with each other. I'm just going to run past these. And so here they are. So just shown superimposed on the DAPI channel by itself. So these are the cells that are in contact with each other. So we can map that out. And this is just a close-up showing those cells. And it utilizes basically an overlap of 3 pixels between the borders of the cells and then a 66th percentile algorithm to positively correlate their association in space. We've moved further to look at not just the contact of cells, and this was like a dream of mine because I want to look at cell dynamics. I'm like a classical cell biologist. I want to look at not just a static picture of where the cells are, but what they're doing as well. So we've also, with the help of the team at TissueGnostics and their app development team, thanks to them, developed an application that can look at the distances of cells to each other. So they have these gradients of distance, so ranges of distance, to be able to look at a process of what I refer to as homing of the cells to each other in a sort of a static sense. We're not looking at dynamically, we're not looking at cell movement, but it still gives a picture of where the cells are in that particular stage of the disease. And so that's a very exciting recent development that we have. This is just the overlay of those contacting cells on the original image. You could, for example, look within follicles. You could look at cell contacts outside and relate that process. This is just another image showing cell contacts within a single follicle. So it's quite powerful. We're starting to look at dynamics of the cells as well as just where they are. The last thing I'll touch upon before I finish is sort of a corollary of looking at multiple markers and having this ability to cross-correlate or register images from multiple scans is to be able to look at sequencing data in cells at the single cell level. and do image-based sequencing. Now, our group, and we have people from MIT already doing this in our facility, they're using just the basic TissueFAXS system, the TissueFAXS Plus, not even the confocal version, as an RNA scope. And so what we've also tested ourselves, and it does work a little bit better with the confocal because you just have a little bit better signal to noise, is to do single cell image-based DNA sequencing. So we have 4 fluorophores for A, T, G, and C, and we do a single pass, and then we haven't done the full set yet because we have to basically develop with TissueGnostics a way of get the XY information between images. But basically, it's building a map. You're adding these markers and building up a map of the sequence and where that sequence is within the cell at the single cell level. So this image is an example of the DNA sequencing work. And then this one here is the RNA scope work. Now, this one in particular is from brain tissue. So this would be from cortex or thalamus or something, but it gives you an idea of the RNA scope work that we're doing. This is just another example that I saw from a user. So it gives sort of a hint at the potential of the system to not just look at protein markers, but we can start to look at sequencing information. We're going to be collaborating with the Broad Institute here to get that sequencing information into their workflow and do it in a spatial context within the tissue. So with that, I'd like to just thank again Rupert and TissueGnostics for inviting me, and also Dr. Naoki Kaneko and Professor Shiv Pillai from the Ragon Institute, who have been great collaborators with me on some of the multiplexing work. Of course, the TissueGnostics software development team, and they're always really helpful and approachable and a very friendly group. Nice to work with over the years. Of course, thanking the Ragon Institute. We're with the Massachusetts General Hospital. We're also associated with Brigham and Women's, funded through the HHMI, and Harvard, and associated with Harvard Medical School. And thank you for your attention.